Am I the one? Yeah, we can.
Hello, everyone, and thank you very much for being with us today. It is my great pleasure to welcome Romain Villoutreix. And for those of you who do not know him, Romain's research interests are focused on understanding how the genetic architecture of adaptive traits can evolve. And he typically answers this question using a combination of approaches, including experimental setups in the lab and in the field, along with very impressive long-term genomic and phenotypic data sets. He studied his PhD at the University of Lille, where he worked on the spatial and temporal adaptation of Arabidopsis thaliana. And after graduating at the end of 2013, he moved to the University of Sheffield in the UK to work with Patrick Nuzzle on the mechanisms of speciation in domestic insects. Since at the end of 2018, he's been based here in Montpellier at CEF, where he's been working on the gene interactions underlying fitness and how recombination and large-scale mutations underlie cryptic coloration, and the role of chromosomal inversion breakpoints on the evolution of supergenes. And today he will tell us about his very latest research in this group, where he will look at what determines whether genetic or phenotypic variation exists in a continuous or discrete state. And we're all very much looking forward to hearing about it. So please go ahead, Romain.
Thank you very much, Arena, for this kind of introduction. Je vais dire quelques mots en français. Je sais qu'il y a des gens m'ont demandé de présenter en français, mais je vois que dans la salle, il y a des gens qui ne parlent que anglais. Donc, du coup, j'avais préparé mon talk en anglais, donc je vais parler en anglais. Je m'excuse. Pour les gens qui ne sont pas confortables avec l'anglais, n'hésitez pas à m'arrêter et poser des questions. Et je peux redire ce que j'ai dit en français. Et maintenant, je vais parler en anglais. So for English speakers, don't hesitate to stop me and interrupt me during the presentation if you have any questions. And so I will start. So thank you very much, Serena, for this kind of introduction. So what I'm going to present you today, I don't know if people hear me online well, or if I need to speak louder, but don't hesitate to say it. So what I'm going to present today is a result I and other member of the team got, and that we are trying to publish. So if you have any suggestions, or if you don't understand something, it means that I didn't explain it clearly enough. So let me know. That would be a huge help for us. And so obviously I did not do this on my own. So that's the other co-authors that helped me a lot put those results together, especially Clarissa and Patrick, on a lot of the color crypsis aspects. So when you look at species in nature, you see often polymorphism, and something like these polymorphisms can come into two forms. So you can see continuous variation of a certain trait. For example, here, color of the hindwings in a moth, or this variation can come in a more discrete way. Like I took here, for example, Ecological nemata, where you have different morphs that are very well separated between each other. And so a big question in biology has been to understand why sometimes you have continuous variation, or why sometimes you have discrete forms. It's a bit linked to the field of speciation, where you like discrete variation can ultimately lead to speciation. And so discrete morphs and discrete forms have been have fascinated biologists for a long time, starting with Darwin and Mendel, where Darwin was in his work very interested in a variation in the flower morphology in Primula, which he studied extensively and wondered why this variation was maintained and what was the aim. Gregor Mendel used discrete traits to kind of, I mean, it allowed him to study the genetic mechanism of heredity. And so, and later workers were very much interested in discrete forms, usually because they were easy to, I mean, from the phenotype, you had easy access to the genotype, and that allowed them to try to understand what maintained variation in species. And coming back a bit more to those, Darwin and Mendel, a huge part of the synthesis made by the neo-Darwinian synthesis made by Fisher and previous worker was to understand why. So Darwin saw variation as more gradual and more continuous, and that did not fit with the result of Mendel, which usually saw very discrete and was studying the mechanism, the inheritance in very discrete traits. And so those later workers tried to come up with ideas and explanation on how you could obtain a discrete variation from continuous. One mechanism that Fisher offered, and then Ford kind of extended, is that recombination can evolve actually between loci and form a discrete variation. So from all the past work, we kind of know that a couple of genetic factors can affect the type of variation we observe. So if you have, so if this is a representation of a chromosome with one locus and then two allele, one giving you, I don't know, lighter coat or darker coat. If you want, the number of loci you have can influence the type of variation you have. So if you only have one loci with two allele, you might have less variation, and that will tend to having this discrete variation. Whereas if you have another example where you have multiple loci, and then all with an allele affecting the same phenotype, but at a smaller amount, you will, because of a recombination between all those loci, you will tend to have more individual with intermediate genotypes, and then you will end up with a more continuous distribution. Another factor that come into play is can be ecological discontinuities, and so ecological discontinuities can, for example, if you have, in the case of a coat color, if you have different background that offers different niche to hide, this can generate a strong disruptive selection. And so if you have a trait that is polygenic and have a continuous variation, natural selection, by counter-selecting individual that have an intermediate coloration can, in the end, push for a more discrete variation into of this trait. And so we can extend this concept a bit further with so one expectation you have in nature when you see an ecological variation and then a trait that is associated with it is the stronger or the more discrete this variation is. So the more discrete this variation is, and the stronger the disruptive selection you're going to obtain, and very likely the more discrete your phenotype is going to be. So linking this to past work, so many work, I mean, I took some flagship example of disruptive selection in natural population. So we have So what's known in all those systems, what is more or less known is that disruptive selection or divergent selection is what's maintaining difference in phenotype. So it can be here color of coat or presence or absence of lateral plates on fish, those fish from lake and those fish from the sea, or different sizes of the bill in Darwin fishes. Surprisingly, I wanted to look into the literature for some of these examples. The ecological factor associated with variation is not completely understood. Especially, for example, in the case of the respine sticklebacks, there are still debates on if it's really due to predation, or if it's due to another hypothesis is it could be due to the mineral composition of water that prevents plates from growing. And even in the case of mice, they've done some tests with plaster, but they don't know so well what is the actual avian predator that is affecting this phenotype. And so, because they don't know the ecological factor, most of the time they, with the exception of Darwin finches, where this is pretty well rounded up, but the selection is often inferred, so they don't know exactly to what extent they have disruptive selection. But and more importantly, in all of those examples, you cannot. The studies have been done on a set level of ecological discontinuities, and you cannot test if different level of ecological discontinuities can actually affect your phenotype and increase disruptive selection and affect your phenotypes. Is everything clear? And so that's where the results I'm going to show you in TMMA I think are interesting. So the organism I've been working with for a long time now is TMMA stick insects. So they are phytophagous insects. And they live and reproduce on the plant they feed on. They are wingless, so they don't move much. And so we have, what is interesting about this stick insects is we have 11 species, and they are all restricted to California, a bit Mexico, like we have a couple of species that are, one species that is down in Mexico. And what's really interesting about those insects too is they span, they live in a different range of environment, and especially different vegetation types. So we have a couple of species that are coastal. A lot of species live in a chaparral, more bushy type environment, and some are found in a more alpine setting. And so what's interesting about that is and surprising with those insects is they do use species like plant species of like a broad range of family, going from angiosperm to gymnosperms, which is quite rare for a lot of insects actually. And because they very likely taste good for birds and lizards, they are heavily predated. And so one aspect, nice aspect of those insects is they developed cryptic body coloration. So here is an individual of TMMA bartmani living in an alpine context. And so you can see that it's kind of mimicking the needle pretty well of its white fur here. So. But you also have more brown mimics that kind of emit the twigs or the stem of host plants. So that's a male of TMMA christinae, the species TMMA christinae. And what's really interesting in this system is that you find green and brown morphs within the same species. And they usually live on the same bush. they usually live on the same bush and then they readily interbreed. So here what you see is a male riding a female. And they tend to guard the female.
Maybe I didn't know. Each species is specific to host plant, but the genus live on many genera.
No, so that yeah, so no, that's a good question. Each species has a diverse set of species it's living on. Well, I'm gonna come back to this. So, for example, Timema shumash, this species will live on mountain mahogany, and then like 3 different species that are more bush. Timema bartmanni, for example, it's going to live on different gymnosperm species, but not usually angiosperms. And Timema crescina is also you can find it on 2 species of bush. So that's what also surprising is that the species have like kind of usually 3-4 hosts that are specific to them, yeah, usually, but you find them on multiple hosts actually. And what's interesting is in almost hosts you find green and brown. So that, yeah.
Green and brown are different species or the same species?
The green and the brown, they are the same species. Yeah, so you find green and brown within the same species. So that's actually the slide that comes. So in Timema shumash, for example, so that's something we knew because we already studied them. But when we sampled extensively. Patrick and I sampled extensively Timema shumash in 2015, and what we were struck is that in these species we had a huge diversity, and then color seemed to be more continuous. And whereas in Timema crescina, which is the species Patrick historically worked more on, usually have a green morph and then a melanic morph, but not much variation. And in Timema bartmanni, which lives more in the alpine context, we had green morphs and brown morphs, but not much variation. So, it's something that we kind of knew for a long time, but we never quantified actually what the continuity of this phenotype, and did not link it with the diversity of the plant, which is what I'm gonna present you now. So the expectation in Timema when we saw that is that very likely in the species that shows more variation for and more continuity for color, we expect. So here it's a presentation of green to brown color. We expect to have very likely hosts that also have more diversity in colors, and very likely hosts that select less strongly against intermediate. Whereas in those species where the morphs are very discrete, the expectation we have is that the hosts they live in have very different sets of colors, and that's it's generating a very strong disruptive selection.
Does the same species, does it feed on the leaves and on the twigs within the same species?
No, that's a good question. They all feed on the leaf. They all feed on the leaves.
Rest on the twigs or
exactly. Yes. So they would rest at different places. We don't have much data on resting, and it depends on the stage. It's a bit messy. In adults, it's not the case in juvenile, but in adults, the melanic tend to rest more on the twigs. But it's not. You don't find it for all stages of life. So my guess is they move a lot, and then
because I guess the twigs are brown and the leaves are green.
Yeah, exactly. Usually, but yes, yes, that's usually what it is. But you also have sometimes brown leaves, especially in oaks, or in some Sometimes in fur, especially, you have like dead needles that get stuck, and so that creates more of an heterogeneous, leaf pattern. And so yeah, the first result I'm going to present, the first part is actually verifying that we have different. Quantifying the color variation issue match in terms of discontinuity, and then looking at host plant coloration and see if our predictions are working, which is we expect to have hosts that show more variation of color here, or at least less discontinuous, whereas here we expect very discontinuous hosts. So these are very green leaves and very dark bark. So to do this, I sampled with colleagues population of Timema stick insects, and then I used a previous sample done by other students. But in total, for this work we have 3 populations, and we sampled 881 insects from 3 species. So 3 species I described you. And we took a standardized picture to quantify color in a quantitative manner. So the way we did it is take the standardized picture with the following setting and measure color on the lateral side of the thorax, and then extracting the amount of red, green, and blue we have. And coming with 2 traits, which kind of described color in a quantitative way. So you have the first, and in an orthogonal way. So you have the first trait that's basically describes very well the amount of green over blue. And then another trait that describes very well the amount of red over green. And so, I mean, I placed here insects, so you can see more or less what the color is expected in this space. But so that's usually the 2 color traits we come to. And so. So that's a representation. So what you see here is for the 3 different species. So Timema shumash, Bartmanni, Christinae, their quantification of color represented in this color space. So green, blue, and red, green. So we did a cluster analysis. So the green dots you see are individuals that were in the cluster analysis identified as green, and red identify as brown. And what we did is to study the distances between those 2 clusters to try to see how different they were. And so if you look at shumash, which shows the most continuous variation, the cluster are the ones that are the less differentiated, whereas Bartmanni start to be a bit more differentiated, and then Timema Christinae is very differentiated with cluster quite far apart. So that kind of confirmed our initial hint, just looking visually at those species, that we had actually had different type of variation. Then we went on to quantify the host plants of those species. So that referred to the question you asked earlier, where Timema shumash, we usually find it on oak and Cercocarpus, which is a mountain mahogany. So it's a small bush. It's a shrub. Bartmanni is much more on gymnosperm, so it's a. I'm sorry, I just know the English name. I think it's a white fir, and then we find it on pine. And Timema Christinae, so those are usually big trees, so we collect only the bottom part of the trees. And then Timema Christinae lives more on bush in the chaparral habitat, so that's a rosacea, but it's making a small spine, like needle-like leaf. and Ceanothus, which you can find in ornament here in Montpellier. I mean, it's a different species, but it has blue flowers, and it's very nutritious for insects, so you find a lot of insects there. And so we collected cuttings from 6 species. We took 203 samples and took standardized pictures, the same way than Timema, and then did around 800 measurements on leaf and stem of those plants. And kind of did the same analysis. So that's so up you have the variation in Timema, and at the bottom you have the variation of their host plants. And what is kind of striking is in Chumash, like stem and leaves seems to be forming one kind of big continuum, whereas in Bartmani, some leaves are possibly dead in the middle and then are in some ways closer in color than the stems, but you still see quite a differentiation between those 2 clusters. And then in Christine, you have also quite a big differentiation. I can't see it too well, but basically, I mean, what comes out of this analysis is that, as we predicted, the species with the more discrete trait is also the species with the more discrete host plant in terms of color. And so we're very happy to see this result, but that's for the moment very correlational. And so the next step we felt was to actually verify that the host of Chumash, or some host of Chumash, do generate less disruptive selection than the host found on, especially on Christine, which seems to have the more discrete morphs. And so to do this, we did a mark and recapture experiments on different host plants. And so, because we don't have much variation, especially intermediate coloration, which is very necessary for this experiment, because they are the individuals that are supposed to be the most affected by disruptive selection. We used Timema Chumash, because that's the species that has the most variation, and then categorize them into 3 categories. So green ones, intermediate, which have helio blue or a bit yellowish, and then melanistic one, or brown one. And so we collected more than that, but in the end we collected, I think, 600 insects, but the intermediate are at a lower frequency in natural population. So what we did is enrich for intermediate form in our bushes. And so we used 120 individuals in the end that were released on 2 different treatments. One is mountain mahogany, which is a host where we observed, and we know that it's very likely maintaining a lot of color diversity, because it shows a lot of like various amount of color. And then the host found in Timema Christine, which is Adenostoma and Ceanothus. And so what we did is release 20 of each category on those 2 treatments, and leave them. So we collected them at the adult stage and then released them after having marked them to not confound them with the Timema that may occur on the bush. And then we released them on a bush that were isolated from other surrounding vegetation. We even trimmed a bit of vegetation around to prevent any migration. And recollected them 3 days after. So they were all collected as adults and marked and then released. And then after release, we collected them 3 days after. And so that's what we recollected. So on the on the host with the most, a very diverse color. What we recollected the most are actually intermediate insects, which seems to suggest that it's here a stabilizing selection more than anything else. Whereas in the host, which showed a lot of color contrasts, where we barely recollected any intermediate, but then collected a bit more green and melanistic. Yeah.
How did you mark them?
With a Sharpie pen. Yes, so just on the ventral side, we marked them with a Sharpie, like making a blue, or I think it was a blue or a green dot. And so it's on the ventral side. So when they rest, it should not be seen by the predators. So. Yeah, so the birds, it was very birdy patch too. And so something I forgot to mention is that they got collected and then released on bush. They got collected on bush and released on bush that were very close to each other, like 100 or 200 meters. So there is no elevational problem or kind of adaptation. It's very likely mainly trypsis happening and then bird or lizard predation at the site. Yeah. So our expectations were met on that aspect, but coming back a bit to my introduction is what we actually found is that in Chumash, at least in some of the hosts of Chumash, we seem to have something that looks more like a stabilizing selection, whereas on the host of Christine, there you seem to have more disruptive selection going on. And so that's come back to my introduction, because I'm also usually very interested in how selection can affect the genetic architecture of a trait. We ask ourselves, so what is the genetic architecture associated with those traits in those two species? And then you have kind of expectation for this genetic architecture in Chumash, where very likely the pattern of selection we detected is stabilizing selection. And given the variation we see, we possibly thought we will have multiple locus associated with color. Whereas in Christine and Bartmanni, because of the host plant color, because it's generating very strong disruptive selection, what we're expecting is possibly to see a single region associated with a single locus associated with color, and if you make the assumption that the same gene are associated with color in those sister species, then it means that very likely maybe a recombination evolved between those genes in those species. Which is another slide to say exactly what I said. So what is a supergene? Usually a supergene is so a supergene is usually a combination of it's going to be either an inversion or a rearrangement that is going to lock multiple gene or causal variants together, and then prevents usually recombination at those genes. And what it does and why it's evolved is it basically locks allelic combination together and prevents recombination. And so that's partly why it's been thought or hypothesized they evolve in the first place, is because if you have a disruptive selection and then the intermediates, so the recombinants or the hybrids, depends. That's an analogy you can make, are counter-selected, then some allelic combination are more favored, and then you want to lock those allelic combination together. And so that's what a supergene does, is doesn't prevent totally recombination because it's usually very hard, but it strongly reduces recombination and at a particular locus. And so, yeah, that will avoid the cost of producing offspring that are less fit, actually. And because the region segregate as one locus. So looking first at the species with the most continuous color variation, Timimashumash. So what you see here is association study in this species. So at the time, the only genome we had available was Timema christinae. So all the graphics I'm going to show you are based on a Timema christinae genome. But it's great to allow comparison across species. So here you have in different colors the chromosomes, so 1, 2 to 13. And then here you have a score of association. So it's called a posterior inclusion probability. So it's an MCMC method to doing GWAS. So it's Bayes images. So it's the number of time a given SNP is found to be associated with the phenotype, which is its color. So for example, in I don't know which time we're random, in 1 million or 5 million model, 80% of the time you find this SNP associated with color. And so why a striking pattern? Well, you have association in other linkage group, but one linkage group which is particularly interesting is the linkage group 8 here. With one, here is the zoom on the particular region. And so that's the same kind of statistic, but zoomed out in this particular region. And for the different color traits, so blue is GB and red is RG. But we saw that we had a lot of association in that particular region. And so that's another graph of this region. But and so when I looked at the genes, the potential genes that were in that region, found actually some interesting genes. So we have one gene which is ST, which codes for the protein scarlet, which is it's a transporter of a pigment precursor in homochromes precursor actually, and it's been one of the first mutants described in Drosophila to explain eye color variation actually. So when this gene is deactivated in Drosophila, what you see is that they have more red eyes because the homochromes doesn't come, so the brown pigment is not coming in the eyes. And a couple of other genes. One is UBX here, which is in the same pathway than the gene cortex. And the gene cortex has been is the one that has been described to affect color in a scale in Heliconius, in a lot of Heliconius species, but also in the pecod moss. That's where transposable elements seem to have deactivated it, and it's creating the paper to like the gray to melanic variation. And so yeah, well, like so in summary, we found quite interesting genes in this region, and then we seem to have like, we can't go at causal variance because the data we used here is DDRAD, but we seem to have 6 to 8 SNPs or variants that are associated with color in Timimashumash in this particular region. What is interesting now is we also did the mapping. So it's again using the Timema christinae genome, and here zooming on the chromosome 8, which is where we see the association. So here in Chumash. So if we're looking at this association in Bartmanni and christinae, so it's more striking in christinae is we have this big bulk of association with color. So every time with color. Here is less, I mean, it's clear that there is association here. So it's another here is another technique. It's like single SNP, but basically it's what's represented in the y-axis is the score of association of a particular SNP with a color. And so there is less clear that it's a non-recombining region, but looking at haplotypes, we have little recombination. So this region, where we find very interesting genes in Chumash, we also find associated with color and Barmanni and have reduced recombination there. And we find it's kind of at the breakpoint of this big bulk of recombination that we know is an inversion now in Timima christinae. So from this, it does look like the recombination evolved in those species to lock the color gene together, possibly other genes too, linked with other traits we didn't measure that are important. But one interesting spin in the story is, especially so it's in Timima christinae. So the plot I'm presenting you is again the same. That's the same region where our color genes are associated here and here. And so that's a polarization along there. And then what I'm representing here is the coverage, so the number of reads that are aligned to this region. And so in green, for green individuals in christinae, and in brown, brown individuals. And so what we kind of noticed is, at the beginning, we noticed it because we didn't have any SNPs there and wondered why. And so we have a massive drop in coverage in this region for green individual, but not for melanic individual, which led us think that in the green genome, we have a deletion at one of the breakpoints that in the green allele that just suppress those genes, and that's possibly inactivating the genes for color and making the insect green, actually. And so that's the same plot, but for Barmanni here, so green and brown, and then Chumash green and brown, and here we don't see any sign of coverage loss, making us think that there is no deletion there in that aspect. Yes, so to conclude this presentation, so one interesting part of these results is that the host plant leaf and stem discontinuity seems to drives color morph discontinuity in Timima, and explain very likely why we have more continuous color or discrete color. An interesting result is that it also seems to drive the evolution of rearrangement in this genus of sitting insect to possibly have some supergene evolve there. And so, that's one aspect I like about those results, is that it kind of it can explain how you can have in some ways big change or big jumps in phenotypes, especially in our case, because we propose a mechanism that can, or our results suggest that at least polygenic variation can be converted possibly quite easily in a step, for example, in the deletion, into discrete categories in supergene. And that's, kind of nice in the, because it could potentially reconcile some shifts in ideas about in evolutions, especially some school of thought that say that evolution is very student versus earlier school of thought that say that evolution is more gradual and involve a small number of SNPs, or not a small number of SNPs, but an accumulation of genetic difference, a slow accumulation of genetic difference. And our results are a bit at the margin of that, where we show that you can convert pretty easily continuous variation for discrete variation. And if you have any question, I'm happy take them. And I wanted to thank a couple of people, so I did not do this on my own. Oscar Mira for a lot of work in the lab and sampling. Some Lucy and Josh, undergraduate student, for helping me a lot with the libraries and DNA extraction, and Amaria also, Stumbuck, that helped me with the library preparation in Sheffield back. A lot of support from the computational team, both in Sheffield and not more with Utah, which I cover. And then we had a lot of help from colleagues in California that provided us lab space, alcohol, freezer, liquid nitrogen to freeze the samples, so Todd Oakley especially, and his students, Emily and Maurice. And most of this research has been funded by the ERC, so I'm very grateful for the support. And that's all. Thank you.
I know you already answered some questions during the talk, but maybe there's more from the room or from people online.
Online, okay, yeah.
Thank you. I had a question regarding the relative density of the different tree colors, or what I mean, like, is there an effect, or maybe you can comment on the possible effect of the relative abundance of plants having intermediate colors, and whether it shapes, what are the size of the different fitness peaks? That's what I mean.
You mean in terms of species densities, like plant species densities? Yes. That's something. Yeah, that's something we've not taken into consideration in that work. First, because it's extremely patchy. So, for example, Chumash, some of the population I know, like you have oaks for 200 meters. So there, and that's why we didn't choose oak 2 for our experiment, because the oaks are kind of big, so you release insect and then you cannot catch them. So yeah, I don't know if it's a proper answer, but the answer is, yeah, the results are maybe a bit to be mitigated with, because it's extremely patchy. So you will have places which are pretty dominated by one host, and then you will have other places, and it depends on all the hosts and the species you're speaking of. But for example, taking Chumash into account, some places are just covered in oaks, and then some are way more patchy with like adenostoma and cyanotis, which do disruptive selection, but you have mountain mahogany just nearby. So yeah, I would say all in all, the landscapes hold in terms of disruptive selection. For example, in Christine, the main hosts we see are just disruptive one. In Chumash, we have some disruptive hosts, but also hosts that maintain a lot of variation that are usually nearby. So, but it's difficult to assess. And to answer a bit more your question, the intermediate forms seems to be sometimes rare. I mean, they are there, but they are not at as high density as sometimes very green over brown one. So it seems they may benefit from aposematism, negative frequency dependency selection, which is another aspect of it. Oh, there is a question. So Arnaud S2, maybe Arnaud, you can ask. I'm not sure I understand the question. Maybe you can ask it live if you're still around. So I could see some Manhattan plots peaks on all the region of the genome and the super. Yeah.
Yes, sorry.
Yes. That's true. I focused on. Yeah, so the question is that you could see peaks of association elsewhere than on linkage group 8. that is associated with color. And so he wanted me to comment on that. So I have two answers, and I don't know which one is the good one. We are using sometimes, I mean, for those species, we're using Timema christinae reference genome. Yes, there's going to be 3 answers. So we're using Timema christinae reference genome on those species, but there is some rearrangements sometimes happening. So that might be a real signal, but on a region that translocated from the color locus. And now we have a new reference genome on those species, so we should be able to say this. It could also be misassembly, which happens sometimes quite often. And I had the case with Timema shumash when I was aligning it to the previous reference genome of christinae. It was not at all aligning to the same region in color than the Timema christinae. And then we developed a new reference genome, and then the signal was clear that it was associated in the same region. So it could be misassembly. Or those could be also real, and also maybe modifier loci or things like this. I've always chose to focus personally on this region because since the start, when I started to do GWAS across species using Timema christinae reference genome, I could see a lot of species with a huge block of association on this chromosome. And the only region that was consistently associated with color is this, when we call it the indel locus, because it's an indel in christinae, but what's this indel locus? And my hypothesis since the beginning is there is not a lot of ways to do color. And so that very likely the region that controls color should be homologous in all those species. And so that's why I focused my effort on studying these species in particular. It doesn't change anything. I mean, even if there is modifier loci, it doesn't change our conclusion that some inversion in christinae and then a deletion kind of locked the allelic combination there together. But yes, there could be other modifier loci on other chromosomes.
Did you make some?
Does it answer your question, Arno?
Yeah, yes. But did you make some?
To a computer. I don't know.
Yeah, sorry. Did you make
Does
some?
anybody else have a? Yeah?
Simple. Do you hear me?
I still like to have more information.
Do you hear me?
But I mean, maybe you don't have it. But how long they spend on leaves and stems. I always thought that descriptive selection was like when you have a population and individuals are going to specialize in one environment and the other are not. But when you have individuals that anyway have to go through both environments, like rest and feed in your case, I find it weird that you would have descriptive selection, like some individuals better adapted to your needs and some individuals better adapted to your stems. And
yeah.
I can't see the, the scenario, but yeah, do you plan to have more data on the how long they spend on feeding and how long they spend on
I mean that it's not the first time it came up, so it definitely would be useful to have to know a bit more what they do in terms of behavior. I mean, that's something we've not tested for the moment, but I mean, birds, I mean, it's also, the main predators are birds, and then there is different species of birds in the bushes, at least that I've seen in christinae, and then they hunt differently. Some birds hunt from the bottom, and some hunt from top. And then what's known in birds often is they sometimes develop a search image depending on the, but it's also dependent on insect densities, sometimes on the patch and everything. So I would expect that maybe all this will create possibly some negative frequency dependent selection on the color morph. And so that's why maybe independent of where they are resting, birds are more looking for greens because the greens are very abundant at some point, or then the melanic become a bit more abundant and then they look more for melanic, but we don't have data on this. We know we have negative frequency dependent selection on Christine has 2 green morphs. They have one with a stripe and one without a stripe. And we know we have the negative frequency dependent selection on those 2 morphs. So it's not far-fetched seeing that could happen in other for this color phenotype actually. But yeah, I mean that's one of the comments of the reviewer who's just like, what are they doing? Why is they resting? And I'm like, oh, we don't know. So, but yeah, so it's possibly something to get data on. Arnaud is asking me if we did some simple formal crossing experiment between morphs. Yes, only in Timema Christine, we went to F2 and then we saw that color was it's not me that did it, it's Aaron Kamao. So he has a paper with the crosses that is in current biology in 2015. And so he saw that first that the green were dominant and that the color is controlled by a single Mendelian locus, actually, if that was a question. But they are hard to, the problem with Timema is they are great in a natural setting, but they are very difficult to rear in the lab because we brought them multiple times, eggs, and I'm guessing it's a bacterial thing, is they all die at the first instar when they emerge. So I'm guessing they don't have the bacteria they need or something like this, or the food they need. So most of the crossing we do in the field. So we put male female in a net and then come collect we put the eggs and then come collect them the next year. So it's a bit of work. It's like, well, yeah, to get F1, to get F2 is like 2 years.
So you're sure that there's no plasticity? It's all driven by.
Yeah, that's something I didn't mention. So when I do the GWAS, like the analysis, the first method I use allow me to see with the SNP data what the percentage of variance I explained of the phenotype, and for color. In most species, but Chumash, but in most species, we explain 95% of the variation of color. It's crazy. I mean, when you look at the peak of association, that I was doing GWAS in Arabidopsis, I never got peak like this. Like the first time I saw this, I was like, damn, that's pretty strong. So I would say in most of the species, we explain a lot of the phenotype. In Chumash, which is one of the species we explain the less, we explain 30% of the phenotype with our SNP. Which is still decent. I mean, some people will kill to have that, but in comparison with other species, yeah, I mean, other species there is barely no plasticity, and we don't see them change color. Like when you catch them juvenile as a one color morph, we don't see them changing at least morph. So they may change the hue a bit. And maybe diet is also helping, on that. But definitely if they are green when they are juvenile, they will stay green very likely. And vice versa, yeah.
Maybe it's too technical, I don't know, but like when you say there's an association like 90% or 95%,
yeah,
like it's to the colors and to the real value in the color space, or is it like on your is it kind of a GWAS, on green versus melanic?
No, it's usually on. It's usually. I mean, for Christine, it doesn't matter. It's so correlated that when you do it binarily, it's you have the same. But I did it quantitatively, and yeah. So the value that I was telling you is more quantitatively along Yep, I can come back. It's along the RG axis. I find usually the axis which is the most informative for green brown is this axis, green red over green, and so that's usually the one that shows the best association in terms of color. And so it's a quantitative association. Well, that's why it's pretty strong in that aspect. Yeah, I mean, I could not be, when I was looking at some results, I was like, damn, I explained 95% of my phenotype. It's not bad.
A little after 12, so maybe we'll just finish with the final comments of Arno, where there are so assortative makings between color forms.
Ah, yes. Great question too. So far, Aaron also studied this in his 2005 current biology paper. So the only species where we have data for this is Tinea macrystinae. And what seems to happen is that the melanistic, like the brown insect, are advantage, they have a sexual advantage. But so basically, if you put two melanic together, they will mate preferentially with each other. And then if you put a green and brown, then they will preferentially, and then two green is the least preferential. But so it's not associative in that aspect. It's like the melanic have a mating advantage, like the brown. But yeah, it's likely playing in the maintenance of the polymorphism a bit.
Thank you very much, Romain.
No, thank you. Thank you all.
He will be with us. We'll have lunch at the canteen, and he will be around during the afternoon as well. So please feel free to join us and chat with him. Thank you.
And if you have any comments, don't hesitate to send them to me by email. Or...